myself, this is polar. Give them little legs just like this. So this is what a phospholipid looks like. Not literally, but this is how we're going to draw it visually, so we can kind of get an idea. But this is going to be the makeup of a lipid bilayer. Alright, so these are phospholipids. Okay, and so let's remember again, what we have is a polar side and a non-polar side. So let's say these guys are in water. Okay, what side is going to like working with water? Well, we said water is a polar molecule, right? So what do we say polar was? Positive side and a negative side. So when you have these guys interacting with water, they're going to like water. Okay, so these guys are hydrophilic. Philic meaning loving. And these guys are going to be hydrophobic. Like a phobia. They're going to hate water. So what happens when you put this in water? Well, it can make a circle. It'll make a little fat globule. Or this will happen. It'll attract both sides. So what you have here is two hydrophilic sides and a hydrophobic inside. So H2O doesn't like it there. H2O out here loves it. When this happens, now this can make a big ring, right? It's going to go on the inside is going to be the inside of a cell, and the outside is the outside of a cell. And this is what cells are made of. This is the cell wall, the plasma membrane. So, the problem that we want to solve here is how does a cell get stuff on the outside of the cell to be inside of the cell? Well, it's going to do something called passive diffusion or simple diffusion. Now there's a couple different ways that this can happen. We'll talk about passive first because this requires no energy. No energy in the form of ATP. There's going to be, later we'll talk about active diffusion, where you're pulling something across its gradient, going upstream, if you will. But what we're going to talk about first is this passive diffusion. So, in order to get something from here to here, it needs to be small. It can't be a large molecule. It needs to be small, and it needs to be nonpolar. If we have these guys that are nonpolar, they're going to move right through pretty easily. Okay, so how does that happen? How does that work? Well, as you know, there is a constant movement of atoms. They're constantly bouncing around. And so what you have is, let's say, got a bunch of these guys over here on the outside of the cell. A continuous diffusion, right? Can go both ways. But it wants to be happy. Now, it's not going to go this way and continue going this way. Continue going this way, because there's too much over here. It's almost like when this comes over here, it's going to push one back down. It's going to push one back down. So it's always going to be Nice, simple. So again, it's got to be small enough to get through here, and it's got to be non-polar. If this was water, then this would be called osmosis. So, we talked about osmosis, we talked about the passive diffusion, but now what happens when we have things that are polar? Something like, let's say, sodium, right? It's got a charge on it. How's he going to get through? How's he going to get through this area? Well, there's proteins. What's going to happen is, let's say you have one that's over here, and then he's going to work his way through. He's going to have a helix, and then boop. So this is a single domain type of protein. You can have multiple domains, so comes through again, and comes through again. So this has three domains of a protein, right? And now this part is going to keep it inside this hydrophobic region, and if it tries to push out, the hydrophilic regions are going to push it back in, and it's going to be kind of anchored here. And these parts of the protein are going to be able to react with things on the outside and communicate with that is on the inside. So this is a alpha helix. Put that over here. Alpha helix. There's another form, and that's your beta sheet. Your beta sheet's going to look something like this. So you have our protein, and it's going to create this flat, folded protein, and then burp, protein, and then burp. Right? And you're going to have these going different directions. Right, so you can see the difference in the shape here. Unlike that of the helix, this is a beta sheet. And what this ends up forming inside this lipid layer is a tube, if you will, a channel where things can go in here and come out the other side. It's going to create a hole. So instead of having this hydrophobic region inside here, things can go through here without being affected by the inside of this lipid bilayer. Here we have our protein. It's the beta sheet channel, right? And now let's say we're not working with something like oxygen, which is non-polar. Oxygen's going to diffuse right across that cell membrane, no problem. Now we're working with something that's charged, like let's say sodium. So these guys now go through this channel, go through this one, go through here. Now we have 4 on both sides, and now it's happy. And again, this can happen both ways, but it's going to push this one back down. And this is called a concentration gradient. Concentration gradient. And again, this is still passive because there's no energy that's being expended here. No ATP. It's just moving along this concentration gradient. This concentration gradient is very, very important. In passive, again, this is passive. In order for this to happen, it's always going to move down its concentration gradient. You can think of it as going downstream. It's not going upstream. It's going down. So it requires no energy. So we had just talked about the passive concentration gradient diffusion of sodium moving along, and it was facilitated by this protein. There's another way that I can do it. The last one was called a channel, right? That was our tube. Just kind of move down the tube. Now, sometimes what you'll have is a carrier protein. So this one, he is specifically shaped like a paperclip. So when a paperclip comes in here, it recognizes, causes a conformational change. This carrier will then close, and it'll open up the other side. and it'll carry this paperclip inside. Now, it can happen the other way too, and again, this is still considered passive because it's still working with the concentration gradient. It's not using ATP. What we'll now talk about is how this happens with the use of ATP to push this guy against its concentration gradient to move these guys up here, which is good if you want to get rid of waste or you want to bring in extra sugar, something like that, into the cell where the cell needs more energy, but it's got more concentration on the inside than the outside, but it needs that sugar. How can we get that stuff across this lipid bilayer if it can't come through here naturally? What kind of protein do we need to do that? How does that work? So let's say these paper clips are our sodium, and these magnets over here are potassium. So as you can see, there's a lot more sodium on the outside of the cell, and we want to get this sodium to go against its concentration gradient. How does it do that? Well, you take these little sodiums and you add ATP, or adenosine triphosphate. This is the cell's energy. Okay? This is going to create a conformation change. These guys are going to go inside this protein. So triphosphate. Keyword here is tri, meaning there's 3 phosphates. Alright? So, what's going to happen is one of those phosphates is going to come off, and it's going to turn this into diphosphate. It'll no longer be tri, it'll be diphosphate. And this phosphorylation is going to cause a conformation change where this little protein is going to close up, open up on the other side, and it's going to shoot these guys into here, and it's going to pick up this potassium. And when this potassium comes in here, when this guy comes back into here, back to here, open the other side, potassium's going to come out into here, and now this guy's ready to pick up some more sodium. And that process is going to repeat. Phosphorylation, ADP, this closes, opens up, more potassium is going to come in, and the process is going to continue. This is active transport with ATP in a sodium potassium pump. This is co-transport. Okay, so what we have here, sodium, there's glucose on the outside, extracellular. See here, there's more sodium on the outside than there is on the inside. So in co-transport, you're going to use the sodium to bring in this glucose. But in order to get the sodium out, we have to do our pump. And instead of potassium this time, we're going to send in these guys. Here comes our ATP. Those guys go over here. Inorganic phosphate comes off. This turns into ADP. This guy closes, opens up, shoots these guys out. They're going to come back down their concentration gradient. They want to be on this side because there's too much sodium over here. So these guys are going to fall down here, and when they do, they're going to drag this glucose with it. This is going to close back up. It's going to open. ATP, inorganic phosphate, is going to phosphorylize, cause a conformation change. 3 sodiums are going to come through this protein. Now there's too much sodium on this side, and they are going to grab a glucose on their way in. It's going to move across the concentration gradient. Now what's interesting about this is we're assuming that the inside of the cell has more glucose. So glucose is moving against its concentration gradient. Sodium is moving with its concentration gradient. So co-transport is a type of active transport